jalan yang sampai hanya satu jalan.
Habis ni dah dekat dah ni.
6.
Fresh kan?
So since my foot is swelling, so we are going to...
The basic idea of gene therapy is incredibly simple. It's like if you have a radio and there's a broken wire. You go in and replace the broken wire and the radio works again. Same thing in a human patient. You have a broken gene, and so you put in a normal gene. Cure the disease. So many things that had to be done that had never been done before to try to put a DNA molecule, a gene, into a human being in a safe and effective way.
We're really hijacking millions, if not billions of years of evolutionary progress on the part of viruses.
Good.
You've got defective genes in a person. You've got a virus that can transfer lots of genes very efficiently. Why not combine the two and use a virus in that way?
The theory was they could remove all the dangerous genes in the virus. In their place, they would put a healthy human gene. The virus would then carry this gene into a patient's defective cells. If it worked, the dream of gene therapy would be brought to life.
So at the end of the day, what we have is basically a machine that we can use to transfer any desirable gene into a human cell with very high efficiency.
Miller's team started working with the most promising type of virus, the retrovirus, a virus that can cause cancer. In animal experiments, the retrovirus worked perfectly. But no one knew if it would work in a human.
Another example of promising gene therapy involves muscular dystrophy. Muscular dystrophy is a genetic disorder characterized by insufficient production of a protein called dystrophin. Disease victims' muscles eventually weaken to the point where they cannot survive. Researchers studying dystrophy in rats have successfully used a harmless virus to transmit the missing gene to every muscle of the rat's body, reversing the muscle wasting that characterizes the disease. This is a magnified image of the quadriceps muscle of a normal mouse. The dystrophin protein is shown in green and outlines the cells in the muscle. This is a similar image of the mouse victimized by muscular dystrophy. And this remarkable image shows a muscle in that same mouse 6 weeks after the gene therapy. Protein therapy is similar, but instead of fixing a patient's genes, it is directed at artificially creating the correct form of the misshapen protein and injecting these correctly formed proteins directly into the patient. This may prove to be easier in the short term because we don't have to identify the specific genetic errors that cause the protein to be misshapen in the first place. We only have to supply the proteins that have the correct shape.
Our body is made up of billions of cells. These cells use various proteins to help them perform vital functions, from processing nutrients and eliminating toxins, to regulating physiological reactions and defending the body against infection. Many diseases are caused by malfunctioning or missing proteins in the body. Advances in biotechnology have led to the ability to use living cells to produce proteins to treat some of these diseases. Genzyme uses long-established, proven techniques to modify mammalian cells by equipping them with a piece of human DNA so they can produce therapeutic proteins. The proteins produced by the cells can be used as targeted treatments, or to replace malfunctioning or missing proteins in the body, addressing serious, often life-threatening diseases. The modified cells are grown and carefully frozen in liquid nitrogen for future use. This collection of cells is called a cell bank.